So today we are discussing a new topic of chapter number 5 of botany, which is viruses, and our today's topic is Lytic cycle of bacteriophages. We know that bacteriophage structure is composed of three main parts: capsid, which means head, tail, and the tail fibers. In head, proteinous capsid and nucleic acid comes, whereas in the tail there is a hollow tube which is non-contractile, and outer the hollow tube there is a contractile proteinous sheath which is used for the contraction, which is by the contraction of that sheath the phage genome has been transferred or has been helped to for penetration. And the third part, which is tail, is tail fiber, which are six in number and usually used to attachment on the wall of the host for the lysis of wall. In the tail there is a base plate which contain pins, these are attached with the bacterial wall, with the wall of the cell, and secrete an enzyme called lysozyme, which help in the lysis of cell wall, wall of bacteria, wall of the host, and make the minute holes by which uh, host genome has been penetrated, has been transferred into the host cell. The bacterial DNA is transferred to the host cell. So in this video we are discussing the uh, Lytic cycle of bacteriophage, T-even phage. Uh, we know that there are two types of bacteriophage, two types: T phages and lambda phages. In T phages, they are further categorized: T-even phages and T-odd phages. T-even phages we have T2, T4, and T6. Whether in T-odd we have T1, T3, and T7. There is no T5. And another category we have lambda. So lambda are in lambda bacteriophages. In lambda phages, there is a cycle called lysogenic cycle occurs. Lysogenic cycle. We will be discussing that cycle in another video. But in this video, we will discuss about the Lytic cycle of T4 bacteriophage. So the Lytic cycle is a cycle which is defined as a bacteriophage cause the lysis of cell and killing them. Lysis of cell and killing them. This cycle is also known as master and slave relationship. In this, master is bacteriophage and slave is a bacteria. जिसका मतलब है slave means गुलाम and master means the head, जो पूरी machinery. Why bacteriophage is called master? Because when it is penetrated into the host cell, it degrade the bacterial genome and takes the control of the metabolic machinery of the cell. So because of this, we called master as bacteriophage or phage and slave as a bacteria. In this cycle is completed in 5 steps. The first step is absorption or attachment. Second step is penetration of genome. Third is biosynthesis or replication, you can say. Fourth step is maturation or assembly of the structural parts. And the 5 steps is lysis or the releasing of new phages. Lysis of the wall or the releasing of new phages. So Lytic cycle's first step is attachment or adsorption. We know that bacteria Bacteriophage have tail fibers which are six in number. So, bacteriophage have a tail fibers which are basically six in numbers. These tail fibers contain a binding receptors. Binding receptors are present on the tail fiber which are used for the strong attachment with the wall or of a cell of a bacteria cell. So, uh, these fibers, these tail fibers are strongly attached with the wall of the bacteria on the receptor side of the cell wall. Where the receptors are present, there they attach strongly with the wall of the cell. So this is called attachment or adsorption. The phage attaches to the surface of the host cell. Here you can see the first step is attachment, or this phase is also called as adsorption in some books. So in this step, the phage attaches to the surface of the host cell. The next step is penetration of the genome or penetration. The viral genome means the genome of the bacteriophage, the DNA, viral DNA, enters the host cell. जो वायरस का डीएनए होगा, बैक्टीरियोफेज का डीएनए होगा, वो पेनिट्रेट होगा by its structure into the host cell. So bacteriophage की tail fibers when they attached with the wall of the cell, and there is a base plate जो भी बताया था, उसके पास pins होती हैं. There is base plate, and that base plates contain a pins which are attached with the cell wall, with the wall of the host cell or bacteria की wall के साथ attach होती है, and they makes the minute holes or pores, and these pins secrete an enzyme called lysozyme which is responsible for the lysis of cell wall of the bacteria. जब जब lysozyme enzyme release होता है, that help in the lysis of the wall, and by lysis of the wall the minute holes are created by that pins. uh, when the minute holes are formed, the contraction of uh, contractile sheath occurs. Upon the contraction of the contractile sheath, the bacterial genome has been transferred into the host cell. Means this process is called penetration. जैसे कैप्सिड से जब कॉन्ट्रैक्शन होगी, एक शीथ बनी होती है होलो ट्यूब के बाहर, जिसको हम नेक भी कहते हैं और टेल कहते हैं मोस्टली. तो जब दैट शीथ हैज़ बीन कॉन्ट्रैक्टेड, जब वो शीथ कॉन्ट्रैक्ट होगी, तब viral DNA or DNA of the bacteriophage is transferred by the contraction of the sheath, proteinous sheath. जब उसकी contraction होगी तो वो transfer हो जाएगा, वो penetrate होगा by the contraction, by the pressure, तो transfer हो जाएगा host cell में. So this is the second step of the Lytic cycle. Here you can see कि किस तरह द बैक्टीरियल डीएनए पेनिट्रेट हो रहा है. ये यहाँ से ऐसे आएगा, और ये जो शीट है इसकी मैं बात कर रहा हूँ, दैट दिस इज द कॉन्ट्रैक्टाइल शीट. जब कॉन्ट्रैक्ट होगी तो द genome viral genome is penetrated, will be transferred into the host cell. The third process is biosynthesis, and this step is also known as replication. जब वायरल जीनोम penetrate हो जाएगा in the host cell में, the replication occurs. How replication occurs? In this phase, firstly जब viral genome penetrate होता है, it secrete an enzyme called endonuclease. Endonuclease enzyme degrade the bacterial genome. जो बैक्टीरिया का अपना जीनोम होता है, that genome, ये जो जीनोम है, that is degraded. ये डिग्रेड कर देगा. ये रिलेटेड phage DNA replicates and phage proteins are made. ये डायरेक्ट नहीं होता, इसके दिस अंडर प्रोसेस होता है. फर्स्टली द एंडोन्यूक्लियर एंजाइम इज सिक्रेटेड बाय द वायरल जीनोम. वो एंडोन्यूक्लियर एंजाइम सिक्रेट करेगा उसका डीएनए, और एंडोन्यूक्लियर एंजाइम इज रिस्पॉन्सिबल फॉर द डिग्रेडेशन ऑफ द बैक्टीरियल जीनोम. बैक्टीरिया का जीनोम जब डिग्रेड हो जाएगा, when the bacterial genome is degraded, जो उसकी metabolic machinery of the bacterial cells की होगी, this is now under control the phage. By degradation of the bacterial genome, the phage control the machinery, metabolic machinery of the host cell. When this occurs, जब metabolic machinery under control of the phage uh, start to synthesize its own structural components which are necessary for forming new phages. उसकी जो copies बनती है उस we can say as a word, तो जब उसकी copies बनानी होती है तो वो अपना structure बनाता है to form the new phages. So the bacterial genome पहले degrade होगा by enzyme endonuclease, and जब degrade हो जाएगा तो तब metabolic machineries of the host cell are under control of the phage. Now phage start to synthesize its own structural components which are necessary. After that, at the same time, जब उसके proteinous part बन रहे होते हैं, the double stranded DNA of the phage, of the virus of the bacteriophage is replicated and become doubled. That replicate होता रहेगा और double होता रहेगा. At the same time, when viral genome अपने प्रोटीन के अपने पार्ट्स बनाएगा, प्रोटीनियस पार्ट विच आर हेड, टेल एंड टेल फाइबर. दीज आर द थ्री मेन पार्ट्स, हेड, टेल एंड टेल फाइबर्स. ये तीन पार्ट बनाता रहेगा. एट द सेम टाइम द रेप्लिकेशन ऑल्सो occurs of the double stranded DNA, which is bacterial genome, and that degraded bacterial genome is also used to synthesize the structural components of the new phages. जो न्यू फेजेस के स्ट्रक्चरल कंपोनेंट्स बना रहे हैं बैक्टीरिया, sorry, the structural components which are formed by the phage, degraded bacterial genome is also used to synthesize these structural components. And at the same time, the replication of double stranded DNA occurs and DNA become doubled. Three main parts in this step, three main parts, head, tail and fiber are formed. These in this phase, these come together and form a daughter phage. They form a daughter phage, and in this phase the nucleic acid is also formed. न्यूक्लिक एसिड इज ऑल्सो फॉर्म. जब रेप्लिकेशन होती है, डबल स्ट्रैंड डीएनए रेप्लिकेट हो रहा है एंड बिकम डबल, तो प्रोटीन बनता है, न्यूक्लिक एसिड बन रहा है. तो न्यूक्लिक एसिड विच इज सराउंडेड बाय द कैप्सिड, प्रोटीन स्ट्रक्चर हेड हो गया, टेल एंड टेल फाइबर्स. दीज आर थ्री मेन स्ट्रक्चर्स जो बनते हैं बाय द मेटाबॉलिक मशीनरी ऑफ द बैक्टीरियल जीनोम विच इज कंट्रोल्ड बाय द bacteriophage or the phage. So this step is known as biosynthesis or the replication. Phage DNA replicate होता है, पहले degrade करेगा, फिर replicate होगा and phage proteins बनाएगा. Phage protein बनाएगा जो बेचारे structural components. And the fourth step is maturation, or it is also known as assembly. Maturation or assembly. In this phase, new phage particles are assembled. जो the in the third part you can see that here the head is formed. This is the tail. This is the bacterial genome. Let me mark it out here. See, this is the head. Wait. This is the head. This is the viral genome replication से हुआ. ये टेल बनी, ये टेल फाइबर्स इसके साथ बने. So these three components are the main structure to form a new daughter phage. Then ये बनते हैं, and in the fourth step these all comes in assembly. वो these three main parts come in assembly and attached to each other. And the assembly or the new phages particles are assembled or formed, means the structural components comes together, वो करीब आते हैं, they mix up and form a bacteriophage structure. This is the fourth step, and the last step is lysis. The cell lysis releasing the newly made phages. So the last part is lysis or the releasing of new phage. In this phase, in this stage, the lysis of wall occurs. The newly formed phages, which are formed, जो नए phages बने हैं, जो newly formed phages हैं, these these phages secrete an enzyme lysozyme. They secrete enzyme called lysozyme. By this enzyme, the lysis of cell wall occurs. By the lysis of cell wall, these are released and affect the new bacterium, new bacterial cells. In this step, in the last step called lysis, the matured uh, new phages जो बने हैं वो secrete करेंगे enzyme lysozyme जो help करेगा lysis of the cell wall of the bacteria or the host cell, or by the lysis of the wall of cell, these are released out in the surrounding and affect the another bacterial cells. And at the in single cell 100 to 200 bacteriophages are released. 100 to 200 bacteriophage or newly formed phages are released. So this was a complete Lytic cycle of the 5 steps: attachment, penetration, replication of biosynthesis, maturation, lysis, or the releasing of new phage.